everybody. Now for something completely different, as they say. My name is Ian White. I work in experimental medicine and diagnostics at UCB. And what I'm going to try to do this morning is give you our perspective on how we actually try to establish the case for stratification in drug development. And the example I'm actually going to focus on is systemic lupus erythematosus, trying to get the core question, the key question, is there actually value for patients when we proceed stratification? So I'll start off with just a little background to the disease. Then I'll talk about how we've actually got to this situation where we're trying to address this key question, why we need to address the question. And then I'll start to cover why or how we're actually evaluating the case, we're generating the evidence for furthering the case on for stratification. And I'll talk a little bit about the patient and physician perspective on this, then cover some modelling we've done to investigate and evaluate the payer perspective. And I'll finish very briefly with some commercial considerations and some conclusions. So lupus is a very, very unpleasant, debilitating disease. It's a systemic autoimmune disease. It's estimated to affect between 300,000 and 1.5 million people in the US. And globally, the number of individuals who are actually affected are in excess of 5 million individuals. We've actually got the age-adjusted incidence and prevalence rates there for the US, and they're 5.6 and 73 per 100,000 individuals, respectively. The impact on quality of life with the disease is significant, and it actually remains a real area of high unmet medical need. If you look over the last 50 years and look for innovation in treatment of lupus, it's quite striking that there hasn't been much at all. With the exception of belimumab, which was approved just a few years ago, there's very little new that's actually been brought to the table. So we've got this real imbalance where we've got patients who aren't doing well, desperate for new therapies, and nothing is actually coming to them to improve their treatment and their care. The disease itself, basically it's all about production of pathogenic antibodies, leading to a global loss of self-tolerance and activation of autoreactive T and B cells. And ultimately what happens at the end of this biological process is excessive tissue injury. We see B and T cell signaling abnormalities, we see dysregulation of apoptosis, effective clearance of cellular debris, leading to antibody formation, autoantibodies, and then subsequently organ damage. The diagnosis of the disease and the treatment is actually challenging. It's a very heterogeneous disease. Many symptoms occur in other illnesses and conditions. The nature of the disease, it waxes and wanes. Individuals can present this disease, they can have flare, they can end up in remission for long periods of time without further symptoms actually presenting. So it's a very, very challenging disease for the patients and also for individual researchers, organizations trying to develop new therapies to address this unmet need. So the statement of the problem we were facing is clearly new therapies for lupus are very unlikely to work in all patients. I think this old-fashioned notion of one size fits all is gone. We need to really understand the biology underpinning this disease to be able to better target therapies to patients. Patients, of course, and physicians and payers are all demanding that new drugs are made available. And increasingly they're asking for more information about those drugs. They want to know how it works, in which patients will it work. The patient wants to know how effective will that treatment be for me? Will it work for me? If it is likely to work for me, how likely will I actually get access to that drug? It could be very, very expensive and therefore not affordable for that patient. So how do we start to establish if there is a case for patient stratification? Well, I guess there are four fairly generic questions that you can ask, and I've actually just highlighted those on this slide. First, is the disease that you're interested in heterogeneous in terms of the biology? I think that's very, very clear for us, because I just alluded to some of the some of the symptoms and the challenges with the disease, there is clear heterogeneity in the etiology of the disease. Is there drug response heterogeneity? Do individuals respond differently to the different options that are available for them? And that clearly is the case. There are individuals who do very well on standard of care. There are individuals who do very badly on standard of care, but may subsequently go on to do very well on novel biological therapies such as rituximab or belimumab. The key question in terms of, I guess, from the payer perspective is the economic value of stratification. Is this trial and error approach, which is often used to treat lupus, is the cost of that so high that support of a companion diagnostic strategy, developing a stratification strategy, is actually compelling? And then finally, is it feasible to actually bring a companion diagnostic or some sort of informative test or tool into clinical practice? So, in terms of patient care, we ask the question, does stratification have a positive impact on patient care and health outcomes? And the patient is obviously at the centre of this. We need to address this significant unmet clinical need. We know that complex diseases such as lupus are very, very challenging for design, execution, and completion of clinical programs. And we also know that in our industry, that biomarker science, personalised medicine strategies are not always well aligned with drug development programs. And where they are well aligned, the outcomes of those clinical programs, on the whole, tend to be positive where they're not aligned. We talk to experts outside of the organisation, we talk to patient groups, we talk to physicians, we talk to opinion leaders, and it's very, very clear. We get a universal message coming back that all of these individuals require to better inform individual patient care. We've got to get away from this stochastic, random process where we try one thing, it might work on somebody, then it doesn't work, we try something else. That's not good for patients. And Hilary alluded to this situation where patients, they're diagnosed with a disease, they don't necessarily know much about it, they don't know how it's going to progress, they don't know if there's a therapy available for them which will work. They have this existence which is totally lacking any certainty in terms of how they're going to do it. So they want to remove this certainty, they want to remove this precarity, as a word that's used to describe this precarious existence that they have, and just talk exactly to that. So clearly, we think intuitively that stratification will have a positive impact, but we need to make a case for that. It's not always as clear cut as we would perhaps imagine. We also need to be aware that any tools or approaches that we bring to the table with the intention of helping patients, they will ultimately have clinical utility if we develop them in a clinical trial scenario where they actually have broad translatability into the real world. Will patients have access to these tools, to these kits or tests? It's not necessarily a good idea to have a very sophisticated test which may only be available in one lab. In a small town in Eastern Europe, for example, it would potentially be a blocker to actually getting access to the test and ultimately to a therapy which may actually benefit patients. And then, of course, there's a commercial attractiveness. Is there an incentive to actually work for maybe a diagnostic company to work and develop testing in this area? So we need to work to understand the value and potential impact of implementing diagnostic approaches to stratified patient populations. So to build the case, I guess we require strategies which are better, more capable of identifying the relevant patients and targeting our therapies to those patients. And for lupus specifically, justifying test development and including the format that require performance, clinical utility, where that test would be used in what population hasn't actually effectively been established. So we've had this discussion internally at UCB, and we agreed that there's a need to fully evaluate as far as we possibly can for all stakeholders, and not just patients, but payers, regulators, etc., the impact of developing and making available tests or tools capable of identifying those patients within a lupus population who would actually respond to therapy. So we put the patient at the centre of everything for obvious reasons, and that's a message that we get clearly from the top of our business right the way down the organisation. We look after the patients, everything else will look after itself. But we have to be mindful that there are other stakeholders who have to be respected, we have to meet their needs and be sensitive to their needs within this overall ecosystem, as it were. So we've talked about patients and physicians. They require tools to better inform prescription and the likelihood of positive clinical benefit. Similarly, we want to know if the test is likely to prove negative for a patient. We want that to be there so we can actually give them the option to access other possible opportunities for treating their disease. For the pharma company, of course, we believe that if we look targeting the therapies to the right patients, we will get better outcomes, we'll get increased uptake, the revenues will look after themselves. The regulators, they have a different set of questions and concerns. They want evidence supportive of efficacy. And if we go to them with a very heterogeneous disease population with limited evidence of efficacy, they're going to want to understand, they're going to want us to demonstrate to them that we understand what subpopulation of that overall population is likely to respond to that therapy. And of course, any tools that we develop will need to meet regulatory requirements before they can be marketed and implemented and brought into clinical practice to help patients. And then the payers, they have separate other questions. For lupus specifically, standard care is actually remarkably cheap and affordable and accessible. Whereas the newer biologic therapies, which are in development with a number of organisations, they're typically very, very expensive. And there has to be a very strong case made to justify prescribing those medicines for those individuals. So as I say, we look after the patients, but we have to make sure that there is this balance and that we meet the requirements, the expectations of all of the stakeholders in this rather complicated ecosystem, as I described it. So how have we actually started to address this thorny question? Well, pharmaceutical companies are typically very comfortable going down the health economics route to establish value for stakeholders. And what we've tried to do, and we've worked very closely, and I'm grateful to colleagues at the Office of Health Economics in London, Adrian Towns's group, particularly Grace Marsden and Martina Garrard. They've run a health economics analysis for us, and we've sought to use this health economics to evaluate what value can be brought to patients. And I'll talk a little bit about that. There were three elements to the analysis. There was a patient and clinician perspective, largely to try to get that patient perspective rather through the lens of a group of lupus physicians. Then we did some, our colleagues at the Office of Health Economics did some modelling to evaluate the payer perspective, and we did some commercial modelling as well, but I'm not talking about the latter today, just for reasons of brevity. So the first element of the analysis was to try to get this patient and clinician perspective. And what we did, we identified a group of 5 industry experts with relevant lupus experience, and we asked them a whole bunch of questions to try to get their perspective on a number of key elements, such as, obviously, what is the current treatment pathway for lupus patients? Is it fairly consistent globally? Does it vary a lot? What is the likely impact if a new biologic treatment was brought to the market? How is that likely to impact the treatment pathway? How would a test to predict treatment response, how would that be likely to support clinician and patient decision making? And how would such a test influence the overall treatment pathway? I think those are some of the key questions. In terms of how would patients get access to such a test, such a tool, typically the current treatment pathway, I've got 3 options here. On the left of the slide, the current treatment pathway is as follows. Individuals will be diagnosed with the disease. First-line therapy is typically antimalarials such as hydroxychloroquine. Depending on how well individuals do on hydroxychloroquine, then there is a fairly well-established route through corticosteroid use, through to immunosuppressants, and ultimately to biologics such as belimumab and possibly rituximab. From a patient perspective, it seems very, very straightforward. I think if I was in the unfortunate position of being diagnosed, I think I would want to get access to tests pretty quickly to establish if there was a therapy that was actually going to help me get to a positive outcome. If there was a good treatment for me, I'd want to actually take that test as quickly as possible. And ideally, I think I would want that on day one. But typically, when we actually spoke to our physician experts, they seemed to recommend, and they were fairly comfortable that that test would be available after failure of first-line therapy within the first year following diagnosis. In the real world, there was also some conflicting views around whether it would actually be used after failure of first-line therapy, and there were some strong views that in the real world, from a physician perspective, the test may well be available once individuals have basically progressed significantly down the care pathway. They haven't done well on antivirals, they haven't done well on steroids, immunosuppressants aren't doing anything, and they're in bad shape. And at that point, the test would then be made available. From a patient perspective, I think the former rather than the latter is clearly the message that we would expect to receive. So very high level results from this first part of the analysis. It was very, very clear, not surprisingly, that an accurate test would increase patient and clinician confidence in a new biological therapy. And there was a fairly clear consensus that high accuracy was needed, and was remarkably consistent. The consensus around a negative predictive value of 90% or greater and a positive predictive value of 75% or greater. Avoiding false negatives was a consistent theme that was raised. Physicians were very clear that they didn't want to actually exclude patients from accessing a new drug if they had a test that could possibly be a false negative. So that was very, very clear. Clinicians were not worried about giving new therapies to non-responders, and interestingly, as was alluded to in the previous talk, patients are becoming more and more involved in the decision-making process over time. But when we talked to physicians in different locations in Europe, we found there was quite a spread in terms of the engagement in that decision-making process with different patient populations in different territories. So for phase two, the payer's perspective, typically we use cost-utility analysis to compare the value of alternative interventions that have different health benefits. So we'd be interested in this case to look at the extra value that the compounding test with a new biologic would actually bring. And typically in healthcare, tools such as cost-benefit analysis, cost-effectiveness analysis, etc. are used. The choice of techniques obviously depend on the decision that you tend to influence. And I think most of you will be familiar with the quality-adjusted life years term to measure health. The primary outcome of the model that we did is actually the cost per quality, or incremental cost-effectiveness ratio, which is calculated as the difference in the cost of the two interventions divided by the difference in the expected qualities produced by the two interventions. And that's the key metric we're hoping to get at, and then relate that back to how that will impact patients. Results of such analyses are compared with a threshold incremental cost-effectiveness ratio. And typically interventions with an ISA below the threshold would tend to be funded, whereas those with an ISA above the threshold would tend not to be in a specific given location. So the modelling, basically the cost-effectiveness model was built to calculate the incremental cost per quality gain of the test plus the novel therapy compared to the novel therapy alone. And what we did, we built a decision tree to estimate a proportion of patients with each test outcome and the resulting treatment decisions. And just very quickly, we have two strategies. We have strategy one, where there is no biomarker, individuals are diagnosed with disease and they will actually be assigned treatment with the novel therapeutic or the standard of care, so they're hydroxychloroquine, etc. Strategy two, we have a test in place. Individuals will actually get a positive or a negative result. The positive folks will be, that group will comprise individuals who will have a true positive or a false positive result. Both of those groups will go on to get the novel therapy. The false positive group will ultimately go down the standard of care group because they won't get benefit from the therapy. The individuals who receive a negative result from the biomarker test, they will comprise true negatives and false negatives, and they will get standard of care treatment of their disease. To build the model, I'll not dwell on this, but we needed to put some data in there. And as a proxy, we built a lot of this work around available data that is used or generated from the belimumab trials. So the population characteristics, we accessed those from the BLISS 52 and BLISS 76 belimumab trial populations. We had to put in some estimates around the test, the characteristics of the test, and we started with a test for the cost of $100 US, which is routinely the sort of area we would expect to be used in clinical practice for a minimally invasive test. We inputted some NPV, PPV values, and the estimate of prevalence, which in this context is the proportion of people who respond to therapy. We had to input some data around treatment decisions. Strategy 1, more biomarker, we estimated based on European market forecasts in this decision resources document, which is referenced here. For strategy 2, the assumption was that patients with positive test results receive the novel therapeutic, and those with negative results don't. We estimated treatment costs and health outcomes, put those in, and also estimated disease activity costs, cost of organ damage, etc. And as is typical in these types of analyses, the costs and the qualities were discounted at 3.5% per annum in the models that were built. These are some of the calculated input parameters when we put all of those figures into our very large Excel spreadsheet. We estimated the qualities for the various treatments, the costs. We had to make a lot of assumptions. There's a lot of proxy data here. The increase in qualities that a responder receives compared to a non-responder is actually the same in this model regardless of treatment. We assume a binary cutoff for the test. Both the cutoff for the test is positive, the lower cutoff is negative, and that determines whether the individual will get the therapy. And for calculation of the costs, patients who get a negative response from the test will receive standard of care or potentially a different B-cell inhibitor. So to cut to the chase, when we calculate the incremental cost effectiveness ratio, that's the difference in costs, or the difference in qualities for these different interventions. What we actually found was that the incremental cost effectiveness ratio was in excess of the threshold that is typically targeted by NICE in the UK. So NICE typically specify £20,000 to £30,000 per quality gain. Where we actually look at, okay, with a heavily caveated model, when we look at the implementation of a biomarker to influence and improve clinical practice in lupus, we find that the incremental cost effectiveness ratio is actually above that. We have clearly a challenge in terms of a conflict between the potential payer and what the clinicians and the patients are actually asking for. So basically, the use of the biomarker is not cost effective as this model is currently at the moment. It may well be by other agencies, but not by NICE. The bottom right is just the cost effectiveness plane, which is a very standardized way of presenting data at these times. So how can we actually bring down the cost effectiveness ratio? How can we actually ensure that what patients need, we can actually provide that? Well, we looked at sensitivity analysis to see what factors we can play with and how flexible we can be with our model to actually influence that ICPR value. So we looked at a number of things: increased rate of response to the therapy, variation in the test accuracy, variation in the population, etc., etc. And you can probably envisage that if you were to raise the bar to find a more responsive therapeutic group, it might be smaller, but with higher likelihood of better clinical response and outcome, that may well affect the ISA. And we've done that, but it's still largely work in progress, I would have to say, so I don't have anything further to say on that. A few points for consideration. I guess another way of saying what I've already said. The model implies a new therapy will need to produce higher quality gains than belimumab for lupus treatment, or for the test and therapy in combination to be cost effective is the current point threshold for current practice. Whilst clinicians require the NPV of the test to be high, this actually wasn't a key parameter for determining cost effectiveness. The positive predictive value of the test work was actually more influential in terms of influencing the ICD. The population, unsurprisingly, and the position of the diagnostic in the pathway is actually critical for determining whether or not the diagnostic is cost effective from a healthcare perspective, but that is another story that maybe we could, haven't gone into today for reasons of time, but we could maybe explore that on another day. So just to close, I think it's becoming clear to us, certainly when we have these discussions with our project teams, that stratification has to be tailored to individual circumstances and individual disease indications, and we have to be respectful of the needs of all of the stakeholders. Personalised medicine is coming to the centre of things in drug development programs. We focus very much on the patient. It's very clear that we have to establish the balance and make sure that what we need for patients is actually achievable and meets the goals, the objectives of the other stakeholders in the ecosystem, as it was. So I'll stop there and I'll be happy to take any questions.